invite Dr. Sandhya V, who is going to talk on the whole exome sequencing of recurrent acute pancreatitis and chronic pancreatitis patients, lessons and education. Another possibly fascinating talk.
Thank you so much. I'm filled with a bit of trepidation in giving this talk. I do not study the pancreas regularly in the lab. I'm more interested in Crohn's disease and inflammatory bowel disease. But as I think many of us in the audience would know, Sudipto is an extremely persuasive person. And with a fellowship that I'd received from the India Alliance DBT Wellcome Trust, the aim was to foster closer interactions between basic researchers and clinicians, which I think is something that could be better in India, let me put it that way. I think it's taken for granted in most of the countries around the world, but in India we're still trying to bridge that gap between basic research and clinical research. Now the fellowship was mainly aimed to try to foster interactions between my parent institution, which is the Indian Institute of Science in Bangalore, and Christian Medical College Vellore, where obviously we have a wonderful set of clinicians who are deeply interested in understanding the basic biology of the disease phenomena that they work with. So I'll talk to you about two studies that we've done along with Sudipto and Ruben Thomas Kurian here in CMC. It's been a really exciting time, and I think we've learned a lot, and that's why my title is Lessons and Education. I really got educated in pancreas. Now, of course, this slide is more for me than for anybody else in the audience. Nobody needs to read this slide, but I need it because it brings into context some of the stuff that I'll be talking about. Now, as basic researchers, one of the questions we always ask is, what are the molecular mechanisms that underlie pancreatitis? And of course, we've got this beautiful work where we have the fine proteases and anti-protease balance that seems to regulate the destruction of the pancreas. We have a number of gain-of-function mutations that have been reported in PRSS1 majorly. Of course, Dr. Whitcomb is there. Calcium has been shown to play an important role in regulating pancreatic function, either by the activation of proteases as well as regulating premature activation of PRSS1. So we have a few examples of a molecular understanding of what causes pancreatitis. And so when we talked to Sudipto and Ruben at that time, they had identified a family here in CMC Vellore who were their patients, where there was an example of autosomal hereditary pancreatitis across 2 generations. The father and both the 2 children were affected with pancreatitis. This seemed to us a very good beginning as where we could start to try to look at what could be underlying the genetic basis of hereditary pancreatitis. And so we did whole exome sequencing. And what we found amongst all the pancreatitis-associated genes that have been reported so far was a mutation, I'm sorry, there's a fly that's bothering me, a mutation where there was a 2 base pair deletion in the TRPV6 gene in the patients. This 2 base pair deletion was absent in the mother, in the father, and it was present in the father and the 2 children. Now, this 2 base pair deletion would naturally lead to a frameshift mutation, resulting in about 160 amino acids of sequence that was completely unrelated to wild-type TRPV6, leading finally to a stop codon and premature truncation. So if we align the sequences of the wild-type and the mutant TRPV6, you can see that the mutant variant found in our patients was truncated at this position. Now, TRPV6 is a calcium channel, and we know that the mutation now lies in what is predicted to be the ion transport domain. This is a putative structure of the TRPV6 channel, and the region that would have been deleted in these individuals would be the slightly lighter shaded region, which is actually the channel region. So therefore, we would predict from this analysis that this protein would be non-functional. So we cloned and expressed the wild-type variant of TRPV6 as well as the mutant we had in HEK293 cells. In the upper western blot, you can see that the wild-type TRPV6 mutant is predominantly expressed in the membrane, and we were unable to detect any membrane-associated form of the mutant protein. However, if you look at the cytosolic fractions, you can see that the wild-type TRPV6 is also seen in cytosolic fractions, but the mutant is detectable in the cytosol. This would mean that it would not be localized at the membrane, which is where the calcium influx, etc., has to happen. We therefore obviously asked, is this protein functional? Of course, our prediction would be that it was not, but we proved this by measuring the calcium uptake in cells expressing wild type and mutant TRPV6. This was with a classical Fura-2 ratio assay, where cells were cultured in the absence of calcium in the medium for a period of time. You can see that basal levels of calcium were unchanged, whether the wild type or mutant protein was expressed. But when you switch the medium to contain 2 millimolar calcium, which would allow the influx of calcium through TRPV6, you can see that while the wild type TRPV6 allowed calcium influx, the mutant protein that we were working with did not. And so, therefore, okay, so we have a non-functional TRPV6. Now this is where, as basic scientists, we start asking questions about whether we could try to correlate some of the levels of expression of TRPV6 with animal models that we routinely use in the laboratory. Now, we've had a lot of discussions in the morning about the cereulin-induced pancreatitis model. We've modified the protocol a little because we weren't interested in really inducing full-scale pancreatitis, but only the initial stages of pancreatic dysfunction. And so we achieved this by providing 3 injections of cerulin and harvesting tissue after 3 injections, where we believe there would have been some pancreatic disorder initiated, not necessarily leading to the extreme discomfort that you see in some of these mice after 5 or 6 injections. At the end of this time, we looked and confirmed that there had been an onset of pancreatic dysfunction by measuring levels of lipase as well as amylase, and indeed we saw increases. The tissue was damaged with the classical phenotypes that are associated with pancreatitis, such as the lumen expansion, etc. And then we asked, now what is the correlation with TRPV6 here? And so what we did was we did RT-qPCR to check TRPV6 levels without and with induction of pancreatitis by cerulin. And you can see that the levels of TRPV6 are increased during pancreatitis, suggesting that it is the initiation that is actually driving much of TRPV6 function at a later stage in the disease. So this is our model, which is nothing terribly profound, except the fact that if TRPV6 is non-functional in the ductal cells, and TRPV6, I must mention, is a ductal cell expressed protein, we find that the calcium levels will no longer be in. There would be an accumulation of luminal calcium, which could now result in all the other complications that are associated with pancreatitis. So this was the first study where we reported a novel TRPV6 mutation in an Indian family. And the question then was, were there any other variants that were associated in this, which were identified in these patients? Now, these are other variants in Cas-R and FGF23, but you can see that the allele frequency is not rare. It's not rare. It's a common occurrence. And I'd like you to look at the last column, which is the indigene minor allele frequency, which is actually data gleaned from about 1,000 Indian genome sequences that are available at that time when we did the study. You can see that these 2 variants are not rare in the Indian population, but nevertheless, these 2 genes have been associated with pancreatitis. And I'd like you to bear this in mind because this might have relevance in the later part of my talk. There was a copy number gain in the initial segment of the CBS gene, but we didn't investigate that further. This was also seen in the affected members of this family. We then asked, could we now provide a more integrated or holistic picture of chronic pancreatitis by mapping in multiple variants in disease genes in pathogenic pathways, into pathogenic pathways? And this multigenic paradigm of chronic pancreatitis has sparked research in recent years. I think Dr. Whitcomb touched upon it extremely well. So what we decided to do was we had with us actually upward of 70 patients, but we did whole exome sequencing of about 31 patients. These were all patients of younger age, and so we will hope to look for a genetic predisposition in this population. 21 were classified by Sudipto and his gang as idiopathic CP, and 10 with idiopathic RP. Now I'd like you to spend a little time on this table because again this alludes to what Dr. Whitcomb spoke so eloquently in the beginning. We focused on looking at genes that were associated with pancreatitis in individuals. I wish I had a point. Oh yeah, fine. So in this you can see these are all the individual patients that we looked at. And these are the variants that we observed. Those variants which have dots next to them are reported variants which have come from studies outside India. You can see that many of these variants occur in heterozygous as well as homozygous condition in the yellow and blue dots. You can also see that many of these variants actually have quite a high minor allele frequency, which means that they may not be causing the disease. But nevertheless, they are seen in patients that are reported with chronic pancreatitis. This is a distribution of common and rare variants that we identified in this panel. Another thing I'd like to note here is that some of these genes are expressed in acinar cells and some of these genes are expressed in ductal cells. Another important factor that we noted was that there were no discernible genetic differences seen between recurring acute pancreatitis and chronic pancreatitis. So it seems to be a spectrum of the disease rather than two separate disease types. A couple of the rare variants that we observed. One was a rare variant in CFTR. We know that CFTR is a chloride and bicarbonate channel. And this rare variant appeared in the nucleotide binding domain of CFTR. You can see that this is highly conserved across many mammals, suggesting that this variant could have an important role to play in the functioning of CFTR. If we look at the crystal structure of CFTR, we can see that the variant is here in the nucleotide binding domain. The novel variant is here. Some of the patients had a second variant, and this interestingly has been associated with congenital bilateral absence of vas deferens in an Indian patient. So we all know that CFTR is important in epididymal function as well, and so therefore one of these patients had these two mutations together. Another rare variant we observed was in human chymotrypsin C, again a highly conserved residue across all mammals, and this mutation is adjacent to the active site of CTRC. Now, this variant again is a known pancreatitis-linked variant. Whether it's the cause or the linkage is really perhaps what I'm trying to emphasize here at this point. And finally, a novel CTSB or cathepsin B variant. This variant, identical variants were shown or is seen in two patients, unrelated. Again, the residue is conserved, and if we look at the crystal structure of cathepsin B, this is where the variant lies. Now, this is a very interesting variant because this residue has been shown to be ubiquitinated in cells, and we know that ubiquitination results in protein degradation. So the mutation of lysine to a glutamate at this position would prevent the ubiquitination, thereby stabilizing cathepsin B in the cells. Now, one of the things that we wanted to emphasize here is that the variants we saw across our patients were variants in genes expressed in both acinar as well as ductal cells. We looked at single-cell RNA-seq analysis from the pancreatic tissue, and you can see that many of the variants that we observed, many of them are expressed in very specific cell types and multiple cell types in the pancreas. And therefore, the hypothesis we brought forward is that altered function in not just the acinar cell or not just the ductal cell, but actually in both cells could lead to chronic pancreatitis. We looked at the genes we had and we tried to integrate them into a holistic signaling pathway using ingenuity pathway analysis. And you can see that many of these genes impinge on pathways such as homeostasis, inflammation, cell viability, ER stress response, all of these known to be involved in the actual malfunctioning of the pancreas at various stages. And therefore, in summary, only half of the CP or RAP patients have a rare variant, suggesting that common variants could be drivers of the disease. This variant in CFTR, for example, is present in about 50% of the Indian population and has little effect on its own, as shown by other groups. But how, combined with other mutations that we've observed in these particular patients, this could actually lead to the disease. And therefore, we propose this two-hit hypothesis here, where the first hit or the first variant makes acinar or ductal cells vulnerable to environmental stresses or their functioning. And the second hit, which could occur in the acinar cell or the ductal cell, could now overcome the protective mechanisms and therefore initiate the inflammatory process. And we believe that it's a cumulative accumulation of a number of variants which ultimately leads to the disease that is seen in these patients. And we'd like to emphasize that the significance of co-occurrence of genetic alterations in ascena and ductal systems in chronic pancreatitis has been suggested in an earlier study. So I'd like to conclude by acknowledging the people who did the work. A postdoctoral fellow, Dr. Idris Shah, who, as you can see, is in beautiful Kashmir right now, where he's continuing his research. Hari Prasad was also an early career fellow supported by the DBT Wellcome Trust. He's now in the Center for Brain Research in Bangalore. As I said, the funding came largely from a fellowship that I had from the India Alliance DBT Wellcome. And of course, none of this would have been possible without the drive of Sudipta and Ruben in being able to get together all our patient cohorts. And I'd like to acknowledge funding that I have right now from NIH and a postdoctoral fellow in my lab, Dr. Rohit Siddharth, who's going to continue some of the work that we've started in pancreatitis, and the veterinarian, Dr. Harini, who really is indispensable for much of our research. Thank you very much.
Thank you, ma'am. That really wonderful talk, sharing your research. Could we have a comment, Dr. Whitcomb?
I have hundreds of questions, and we'll talk later. Beautiful work, beautiful presentation. I would submit that you know more about pancreatitis than most gastroenterologists. But I think the theme we're seeing is a burden of mutations that change the threshold for causing pancreatitis in some patients. So either have a lower threshold or stronger injury signals, and or a combination of those. And beautiful work. We know that the CFTR SPINK combination is very common, and there may be some action inside the duct itself, which is very important, I think, in India. But beautiful work and a beautiful presentation.
Thank you so much. I mean, coming from you, it means a lot. Gives me a feeling that I'm still standing on solid ground. My main interest is in inflammatory bowel disease and IBD. So there's a little bit of connection.
That's my secondary interest.
Okay, excellent.